So I just want to welcome everybody to the first RIDE seminar of the 2025-2026 academic year. We're really excited about our speaker today and all of the scheduled speakers to come. So I am Caitlin Fyoke. I am an assistant professor in veterinary clinical sciences and the research manager in the Clinical Investigation Center. And I just want to say thanks to the research office, especially Kinsey and Jessica and Tibber for helping set this up today. If you guys have any thoughts on future speakers, definitely contact me or a member of the research office, and we will figure out how to get them on the schedule, especially for next year. We're already thinking about that. And so today we have a speaker that will be introduced here shortly. I just want to give kind of a brief kind of heads up for our next seminar in the series, which is on September 17th, so just next week, with Michael Gale Jr. From the Institute of Infectious Diseases here at the U. If you could also please take like 30 seconds to do the 2-question survey. There's a QR code on the slide, or there's paper copies on the tables in front of you. That helps us improve. And so with that, thank you for coming, and I will pass it over to Hin to introduce our speaker today.
All right, I'm going to test this mic before our speakers using it. So it's my real pleasure to introduce to you Dr. Yewande Fasina. She's currently serving as a professor of Poultry Immuno-nutrition at North Carolina's Agricultural and Technical State University, known as NCA&T University in Greensboro, North Carolina, a beautiful place. If you haven't had a chance to visit it, I would encourage it. Yewande was born and raised in Nigeria, where she earned her first bachelor degree in agriculture there before moving to Canada to complete her Master of Science and PhD degrees in animal science at University of Saskatchewan. She also spent some time at North Carolina State University as a visiting scholar, and actually that's where she's currently holding an adjunct professorship position. And she also spent some time at Auburn University as a postdoc before she joined NCA&T University. So it's a real pleasure to have her here. She actually has some 50 slides or so. So I'm going to give her as much time as she can to present her fascinating talk. Let's see if we can.
Yeah. So this is this pointer. Okay. Which one is the? Okay, so this just go forward. Okay. Yeah. All right. So good afternoon, everybody. Thank you so much, Dr. Hinley, for the introduction. And it's a pleasure to be here in Minnesota. I know, I'm trying not to say it, but I have to. I know you guys are like #1 with turkey production, and I'm from North Carolina, but we're not gonna worry about that. But thank you for having me. So I thank everyone that made it possible for me to be here today. So with that, I just appreciate the opportunity. Yeah, my voice is loud in itself. It's a mic. Thank you. All right. So it's really a privilege for me to share some of the findings from my lab about the impact of nutraceuticals on gut health and also potential impacts on gut-brain axis in poultry. So, and for me, what I've worked with primarily amongst poultry species, that's broiler chickens, broiler chickens. I occasionally work on turkeys, but primarily I work frequently with broiler chickens. So without further ado, let's go. This is going to be my presentation outline. I have 50-something slides through. It's just to make things safe. I'm not going to spend time on some, and some I will take time. So we're going to first I'm going to give a quick background on some things, like what are nutraceuticals. I know most of you know this, but some of us may not have a clear concept of what those are. Then what are the beneficial effects? Then what are the major enteric diseases of poultry? Then how do we combat diseases in poultry and our concerns about antimicrobial resistance? Then alternatives. And this is where I come in with nutraceuticals, and I've brought in the ones specifically that I'm going to touch on today. So essentially, after that, I'm going to now give you a synopsis of some of the projects that we've done and the results. So we're going to look at, to start with, if I want to know how alternatives to antibiotics modulate immune response, then I must first be able to do two things. First, I need to understand how the pathogen actually affects the immune system in the bird. So we first look at salmonella, then the classical antibiotic that is still allowed in commercial poultry production. We'll look at what is the immune signatures of this antibiotic in the spleen of the bird. Then I start looking at modulatory pathways of that, comparing it to the alternatives that I've evaluated in my lab, such as spray-dried plasma, onion peel extract. And then I move to the second disease that I work with, necrotic enteritis. And I share some of our results on effects of green tea extract, which is another nutraceutical on necrotic enteritis. Thereafter, I'm going to give you a brief insight into the fact that we can no longer ignore the idea or the fact that dietary fat types actually can program the behavior of birds. Yeah. So this is streaming into bird welfare and health. Bird welfare and behavior. Yes. So, and I have some data that I would like to show you just to, okay. I have some data that I would like to show you just to help you and have an idea that, okay, dietary fat type actually does modulate the behavioral modulating section of the bird brain, and how we need to start thinking of bird behavior in terms of nutritional formulation and feed supplements and, dietary the ingredients that you use to formulate the diet. Okay, so, and that will be the end of my sharing data. And then I will then give you my vision of the nature of my collaboration that I anticipate with your department here. And of course, I have to thank the people that got me to this point. Okay, so with that, let's write. Nutraceuticals, essentially, they are simply put, they are plant-based or animal-based products that are in the diet, added to the diet for purposes more beyond their nutritional value. So primarily to provide additional health benefits. And really, they are classified into a variety of categories. So functional foods, dietary fibers, DHA, phytochemicals, herbs, probiotics. So for me, the ones that I'm primarily talking about in this presentation fit primarily into phytochemicals or herbs, both. And then when you talk about fatty acids, or fatty acid types. So fats are known now. It is no longer a misnomer. We now know, it's accepted that dietary fats are actually functional foods. So I would say everything, the dietary alternatives to antibiotics that I'm gonna be looking at in this talk fits into functional foods like things, fatty acids, phytochemicals, herbs. So with that, move to the next slide. Benefits in a nutshell. That there are tons of literature about benefits of nutraceuticals in food animal production. So, but to bring this home to us, since I'm talking about poultry in a succinct manner, I figured this chart summarizes it all. It's looking at Innovo, benefits of Innovo supplementation of nutraceuticals. Here are the nutraceuticals, pretty much maybe different probiotics, phytobiotics, etc. So here are the benefits listed by Bajini et al. So creation of hostile environment to pathogens. So that prevents pathogens from gaining entrance into the embryo. Elimination of receptive sites for pathogens, production and secretion of antimicrobial substances like bacteriocins, competitive use of essential nutrients, like usually they use up the healthy probiotics, the probiotics, they use up nutrients like zinc that the pathogenic microorganisms need to grow. They use it up, so they don't have nutrients. And establishment of balanced microbiota. And I can go on and on and on. So, and the rest are here. And the one I just can't afford not to mention is speedy development and functionality of immune system. So it enhances gut development and development of immune system in addition to these myriads of positive effects. So this transcends actually into when the bird hatches and growing throughout life, if you look at the poultry literature. So that's the one I chose to show you. that this benefit starts right actually in ovo and throughout life, depending on the dosage you use and the stage of life at which you use it and the health status of the bird. Now, to hone in my point about dietary fat, I like this chart that I saw. It kind of summarizes my point. Here is when you have gut dysbiosis. The problem with gut dysbiosis is, now, let me back up one step. We all know that dietary fat, when consumed, and if whatever portion of it gets to the large intestine, it gets fermented by gut microbes in the large intestine. The end result of that typically is production of short-chain fatty acids, okay? And when there is dysbiosis or there is reduction or imbalance in the composition of the healthy microbes in the gut, this is what happens. There is no short-chain fatty acids to regulate this angle that contributes to normal functioning of the brain. And that really affects the quantity and nature, that is composition, of short-chain fatty acids that crosses the blood-brain barrier. And short-chain fatty acids actually play a pivotal role in formation of serotonin, which is very important in the central serotonergic system, signaling mechanisms in the brain. And that's responsible for behavior and mood mechanisms. So that's just an example. I want to keep it simple, okay? So, and here again, you see when the normal gut microflora exists, neurotransmission by neuroactive metabolites, those include tryptophan. Tryptophan is the precursor for serotonin. So tryptophan will be able to cross blood-brain barrier in adequate quantities and enhance serotonin synthesis in the brain, and that affects behavior. But when there's dysbiosis in the gut, you see, you get abnormal neurotransmission by neuroactive metabolites. So there is no more doubt about the fact that dietary fat has impact on brain, serotonergic, the central, that is the brain serotonergic signaling, and possibly bird behavior. So that's one of the things I'm trying to embark on in my lab. Like, is there a classical way to prove this and kind of figure out mitigating measures? Of course, I'm not a trained behavior expert, bird behavior expert. So I'm collaborating with bird behavior experts to do this. All right. So next, it's time to look at, okay, I've talked about nutraceuticals and how they can mitigate, how they have potential to mitigate diseases in poultry, and how they have potential to influence the gut-brain axis to affect bird behavior. So what are the primary diseases affecting poultry? So pretty much most of us here are familiar with these diseases. Enteric diseases, my focus is on enteric diseases. That's why I only talked about enteric disease, because I like to keep things focused. So if you look at it, we have bacterial diseases, parasitic, and viral diseases. So for Viral diseases, you have all these coronavirus, adenovirus, and I'm not a virologist. Dr. Hin is going to be helping me with that. So, and there are parasitic diseases, blackhead disease, coccidiosis. My good friend, Dr. Bilki at NC State, works, I work with her on that, and I'm open to possibilities to collaborate further. And for the bacterial, we have necrotic enteritis, salmonellosis, ulcerative enteritis. Hey, even though I work on these two, I'm always open to further collaborations because I believe two good heads are always better than one. So I believe you got my drift. And yeah, we've been using antibiotics to mitigate and control those diseases. But for the last 20 years or so, we all have been, striving to get off that because it's causing antibiotic resistance and through these mechanisms, because when we were feeding the animals antibiotics regularly as prophylaxis back in the day, the bugs in the intestine gradually became resistant. So if humans eat contaminated products, either poultry meat or eggs, and if they don't cook it well and they fall sick, they get to the hospital. The drugs they give them, if it's one in the spectrum of the ones we gave them, they gave the animals, they're not going to respond to treatments promptly. So the doctors have to go to the extra mile. And some people have actually, passed away because of that. So we don't want, bottom line, we all know we're in the realm of no antibiotics in animal feed. So for these reasons, and we are all continuously being educated by ag extension agents to keep our meat separate from plant products. Don't use meat you used to cut. Don't use the knife you used to cut meat to do your salad so that you don't, contaminate your food, just so that you don't get antimicrobial resistant pathogen into your body. So, bottom line, we want to keep avoiding different ways of pathogen entry into our system. So, we are no longer using antibiotics or other drugs, antifungal. You see, I always tell my students when we say people with antibiotic resistance, I still correct myself, antimicrobial resistance, antifungal, antiviral, anti-everything. So it's not only antibiotics. So now, what are the alternatives? Generally, I already showed you the classes of alternatives that we have. So here are the alternatives, and together, they are nutraceuticals, as I initially introduced them to you. So, and the reason why we are exploring them is to enhance immunocompetence and disease resistance in poultry. Here is also another chart just showing these different classes of alternatives or nutraceuticals. Oftentimes, one chart is not exhaustive. So I like explaining things with multiple diagrams or images. Now, let's get to talking about the research part. Okay, so this one is the second, my number 2 agenda. Effects or mechanisms by which selected nutraceuticals react to intestinal salmonella in broiler chicks. So my first step in that is to first understand, okay, what is the baseline? How does the body of the chicken respond to salmonella? How does the spleen, should I say, how does the spleen of the chicken, the spleen proteome of the chicken respond to salmonella infection? And this short introduction is a no-brainer. Most of you already know about this. Salmonella is non-typhoid, okay? In humans that eat contaminated poultry products or food products come down with human salmonellosis. So now, on the poultry part, salmonella in young poultry is really debilitating. It has negative effects oftentimes. So we need to ensure that young poultry is not affected by salmonella. So, and the objective here is we want to know how the spleen proteome responds to intestinal salmonella infection in chicks. And the idea is that if we do this study, perhaps we will be able to repeat this study with the goal of, harvesting some spleen samples and in collaboration with Dr. Hinn, identify candidate epitopes that will be good to make vaccines. Okay, so this is the experimental design. We got day-old chicks from reputed hatchery, and upon arrival at our poultry unit, we took 20 and just routinely tested them, evaluated them, took their sika, necropsied them, aseptically removed their sika, and tested the sika for salmonella using these procedures. So, and after 5 days, we realized they were free of the marker strain that we are using for our experiment. So we know that our experiment is valid, and then our chicks, the remaining chicks on day 1 were already randomly allocated to 2 treatments, which were control, no salmonella, then salmonella challenged. And each treatment had 4 replicate pens, and each pen had 15 chicks. The CRD design, completely randomized design. So here's a picture of one of my students orally gouging a chick on day 2 of hatch. So, and we ensured the diets met or exceed the recommendations, and we gave the birds ad libitum access to feed and water, and duration was 14 days. So we ascetically collected the sika of these birds on day 3, 7, and 14 post-challenge. And then here are the results. We didn't detect any salmonella in the control chicks, but in the salmonella challenge chicks, obviously we detected salmonella. It's confirming that infection was established, and this infection decreased with age, indicating that the bird's natural immune system was actually fighting the infection. So it will be interesting to see what the spleen proteome will look like on these days. So to do the proteome, we assertively collected the spleen on those days, and we subjected it in collaboration with Purdue University. We subjected it to, they subjected it to LCMS, mass spec, liquid chromatography mass spec analysis. See, they first isolated the proteins and did all their fun stuff. And so in the end, the results we got was that we looked at 2 time points. Between day 3 and day 7, we got a total of significantly differentiated proteins. And between control and challenge treatment, 216. And this column shows upregulated proteins, and the next one shows downregulated proteins, and this row is for day 3, day 7. So certainly, we know we are about to see some differences. And then there was a partial least square discriminant analysis conducted. So, and there's heat map, and then there's heat map and dendrogram, okay, of hierarchical cluster analysis, just showing you that there's a difference, because you can see, even with color patterns, the red color seems to be clustered on the upper left here, and a little towards the lower right here, and the blue too was like kind of diagonally split like that. So all those things just show you indication, simply put, show you indication that there are differences between the control and the salmonella infected. So this is kind of a diagram that summarizes what we found. The red arrows show what was upregulated. So during salmonella infection, cell differentiation is upregulated. Oxygen and energy use is increased. Lysosomal degradation of antigenic proteins and infected cells increase. And there's increase in adaptation of the pathogen to the host. There's decrease in cell cycle progression. Also, there's decrease in RNA processing. Okay, so this is how we interpreted these findings. Bottom line, infection increased the production of energy and oxidants. So it means that, then our data, our growth data showed that in this particular experiment, salmonella did not impact growth performance significantly, but infection was metabolically costly, and energy was diverted from normal developmental processes to fighting the infection. So this is the baseline, what salmonella does. Then we went to look at, okay, antibiotic will save the day. And the antibiotic in question this time is bezitracin, methylene disalicylate. Henceforth, I call it BMD. BMD. So following about the same experimental design, this is the results of the salmonella challenge on day 3 and day 7. So Zika salmonella concentration. And again, as you see, the control and BMD that was not challenged, no salmonella was present. The control and BMD that was salmonella challenged, we can see that we detected Zika salmonella concentrations. However, it was lower in the BMD treatments compared to the corn treatment. And the p-value here, even though I realized I didn't put it on here, the p-value here was less than 0.0001. So again here, we took the spleen, subjected to same procedures as previously described, except that, we better annotated it here. So we extracted proteins, then they subjected Purdue to liquid chromatography mass spec, and list of proteins were made, peptide identification, and then data processing occurred using Proteome Discoverer software. So, and the results again showed that we got overall 77 proteins were affected by BMD, and then the interaction of BMD and Salmonella, 126 proteins were affected. This is at 0.05. At 0.1, when we reduced the sensitivity, of course, we got more proteins. So whether it's 0.5 or 0.1, either way, there is the same observation. So here I've summarized the overall BMD effects for those of you that would like to look at it further, because I believe this presentation will be available. That's why I put in this extra information. So you can take time on your own and look at, okay, overall BMD effect, what was upregulated by BMD, downregulated by BMD. And then interaction of BMD and Salmonella, what was upregulated, what was downregulated. So my point here is that in summary, BMD inhibits processes that are associated with inducing inflammatory responses and bacterial cell wall rupture. Yes. That was, when I saw that, I'm like, ooh, I thought antibiotics, they break peptidoglycan cell wall and everything, the streamline thing that we learn in class, they all don't work the same way. So this was a turning point for me. But guess how BMD works? It hardly does anything to the bacteria, anything significant. The way it affects the bacteria is it actually focuses more on the organism by enhancing protein synthesis in the organism, helping it to recover, promote cell cycle progression, and protein homeostasis. That's what BMD does. So it was at that point that I thought, okay, this needs to continue. I need to understand how how alternatives, what's their mechanism of action. So that led me to spray-dried plasma, investigating effect of spray-dried plasma and onion peel. So I looked at not me, my team, my hardworking students, okay? My hardworking students and research associates, not me. So we together looked at the effect of spray-dried plasma and onion peel extract loaded with bioactive immunomodulatory compounds and oxidants on spleen of salmonella-infected broiler chicks. So just so that you see it firsthand, spray-dried plasma is obtained from the abattoir, or when they are slaughtering swine or cows, because they have lots of blood when they slaughter them. So they collect the blood and then they process it in a manner in which they then they centrifuge the blood, then collect the plasma, and the plasma is passed over hot flash of heat. And that's how they quickly dry, so that the important nutrients that you see does not get denatured, particularly the immunoglobulins. So onion peel extract, the bioactive compounds in them are quercetin, primarily quercetin, and it also contains flavonoids and phenolates. And yes, I collaborate with Dr. Sang, Shemi Sang, who is a metagenomist and food chemist in my university. So it's really well reputed in that area. In fact, we send our diets to him now. Anytime we do any experiment like this, he analyzes the diets so that for bioactive compounds. So these bioactive compounds, they have all these activities. So in a nutshell, what we found in this experiment was that spray-dried plasma, the way it operates is that it decreases inflammation in chicks, and it also enhances immune responses to salmonella. So it kind of responds in the classical way we expect, but it does it in a way that the response is not excessive. For onion peel extract, this is supposed to be E. Yeah, so because we are now working on the powder, just so that we can give this to farmers in a way that is, user-friendly, because the extract is very expensive. And the powder too works great. So we are still working on that. So please, this is E. I probably could correct this and resend it to her, all right? So this, when we looked at onion peel extract on display, we found increased T-cell proliferation and enhanced pathogen membrane destructive activity. That's in addition to promoting gut integrity and preventing pathogen translocation and systemic infection. To be very honest, that's the most potent one I've worked with. So that's what all I have for you on salmonella. Then we have a project going on. This is funded by NIFA SARS. ANUP is on that grant. So looking at green tea. Now, for this work, I'm also looking into the spleen. Yeah, one of my students looking into the spleen, but we just submitted the samples. But I will still explain what we have found so far. So we were looking at effects of green tea extract on necrotic enteritis. So necrotic enteritis, it happens, when it happens, it causes chronic inflammation, poor performance. The mortality is really high, especially if it's a clinical one. The mortality is the classical symptoms. And then intestinal damage and lots of dollars lost, billions of dollars. So, and the principal causative agent is different toxins producing. Now, now I think we are at the stage. Anno, please correct me if I'm wrong. We've gone from net B to TPL. So which one is it now? I know, I know, I'm just trying to be sure that I'm not missing anything, because it evolves, it's a fast evolving thing. So I want to make sure I don't tell them the wrong thing, especially when I have an expert like you and if there's any other expert here, please feel free to let me know. I love to learn. Thank you. So one of the predisposing factors is coccidia. So that's the model we use in my lab. So we just use the Emeria vaccine because I don't want to bring the protozoa into my lab so that there won't be contamination with my bacterial work. So I just use the Emeria vaccine. But in the very near future, I'm going to be having my students go to Dr. Bilkis' lab to do the coccidia and then take it directly to the farm so that we can use real life oocysts. So that's the model we used. And green tea has these bioactive compounds. Everything is like epi something, epicatechin, and gallo something, for the most part. Well, it's got this L-theanine also. So the experimental design, straightforward, completely randomized design, 6 treatments, 5 replicates per treatment, 20 chicks per pen, floor pen trial. And here are the treatments. So we have the control. Now, the ACON, anything A shows you that they had necrotic enteritis challenge. So ACON is control plus NE. Then BMD is the antibiotic plus NE. Then GTX is green tea, the smallest dose, 250 milligram per kilogram, then 500, then 1,000. And this is the pictorial representation of the timeline. So day 1, we started the experiment. We recorded growth, way back, day 7. Then we gave Coxivac 10 times the dose, recommended day 13. Then we switched the diet to graphase, day 14. Then we challenged the birds for 2 days with Clostridium perfringens. And then we collected sample and deletion scores and all that, collected spleen and all the stuff we needed to collect from the birds. So on day 1 post-challenge and I'm just guessing now, is it day 7 post-challenge and day something post-challenge? And then we also did flow cytometry on this day and ended the experiment. So for growth performance, the feed intake was not different for all treatments throughout the study. Feed conversion ratio was obviously poorer for all challenged birds. However, the birds that were on green tea, they apparently seemed to manage better than the control without BMD or green tea. Though that difference wasn't significant, the difference between this and this. So looking at the body weight, we found that the green tea treatment at 0.55, at 250 milligrams, and just that one, at 250 milligrams, actually had better weight than the control challenge. Now, another phenomenon I've noticed is that anytime that we gave these birds a phytogenic supplement, any plant extract, they seem to lose weight, but the pathogens keep reducing. We are still investigating that because we have been weighing the fat pad, abdominal fat pad, and we've been observing that the abdominal fat pad actually has been reducing. Now, the poultry industry necessarily does not want excessive fat pad. So we are working on, that's another project entirely. So moving to the next data for lesion scores, beautiful. This is what we found. This is the control challenge. The difference was very clear on day 1 post-challenge. By, I think it's day 7 or day 5 post-challenge. By day 5 post-challenge, you can see the higher doses remain significantly low, had significantly lower lesion scores compared to the control challenge. So there's no doubt the green tea works. And we did not measure Clostridium perfringens concentration. Why? Because it's not like we use the marker strain. And 2, how are we going to know what we are looking at is what we gave the birds? The real classical indicator is lesion scores, and we also did nitric oxide measurements. But because of time, I was nervous putting that data in here, and it was beautiful. It fell in line with the lesion score. So, but I'm showing mortality. So, it's to be expected. We had significant mortality in all any necrotic enteritis, challenge birds compared to the control non-challenge. That's expected. It means, yay, it was successful. Time, right? We're almost done. How many more minutes do I have? Oh, okay. So I'm going to wrap up now. All right. So, bottom line for flow cytometry, we did flow cytometry, but we looked only at macrophage and B cell. So, bottom line, conclusion. GT reduced intestinal lesions, lowered serum nitric oxide levels, and is good up to 500 milligrams per kilogram. So, what do we can't go into the dietary fat. You can look at that on your own. And bottom line is the future work is to work with Dr. Hinn. Invite Dr. Hinn in November, then develop phytogenic regimens for poultry, collaborate on vaccines with Dr. Hinn, and possibly maybe cross-training of graduate students, postdoc, and faculty. Big thanks to Dr. Priya's office and Dr. Hinley and the department, all of you. Thank you so much for having me. And I thank all these wonderful people. Thank you all. I appreciate it.